Good day, we are the group number 7 and for this laboratory exercise number 8, we will be tackling about the different characterizations of bacterial culture on agar plates, agar slants, and nutrient broth. For the objectives, at the end of this laboratory task, we should be able to identify and distinguish basic features of bacterial culture on agar plates, agar slants, and nutrient broth. Also, we should be able to recognize the advantages and limitations of cultural characteristics in the identification of bacterial species. I am Jaisal Masina and I will be starting our presentation. For startup, let us first distinguish the difference between agar plates, agar slants, and nutrient broth. An agar plate, as a standard solid support material, is a layer of nutrient gel in a petri dish that is used to grow microorganisms in the laboratory, and it is usually utilized when working with fungi or bacteria. On the other hand, an agar slant, or also known as an agar slant tube, is a screw cap culture tube partly filled with an agar mix such as nutrient agar. Bacteria may be grown in this second type of medium if the purpose is to maintain them in a longer-term culture. Generally, bacteria grown on slants will remain viable for a few weeks to a few months and sometimes longer if stored in a refrigerator. Lastly, nutrient broth. Nutrient broth is a liquid medium used for the cultivation of a wide variety of organisms from clinical specimens and other materials. This medium can be enriched with other ingredients such as blood, serum, sugars, and etc. Now, moving on with the characterization of bacterial culture in agar plates. For the first one, pigmentation of the colonies or color. Bacteria colonies are capable of producing different kinds of pigments, creating distinct colors. As for example, Staphylococcus aureus produces a golden yellow color. Rhomobacterium violacum results in purple colonies, and Flavobacterium species results in yellow pigmentation. Colonies may also be described as opaque or milky color, translucent or nearly clear, or appear dull or shiny. Staphylococci are usually opaque, while many Streptococcus species are translucent. Second, the surface characteristics. In terms of macroscopic morphology, colonies may appear dry or powdery such as the Nocardia brasiliensis colony, smooth or glistening such as the Saccharomyces cerevisiae, wrinkled or rugose like the Bacillus subtilis, and mucoid in appearance such as the colony produced by the Bacillus licheniformis. Also, contoured colony that is irregular and smoothly undulating is produced by the fungi Aspergillus nidaulus. Radiate colony with ridges radiating from the center like spokes in a wheel, and example organism is Phytophthora arenaria, and lastly, concentric surface like some Vibrio species. Next, we have the various shapes of colony, namely punctiform, circular, filamentous, rhizoid, spindle, and irregular. For the first shape, the punctiform that are colonies under 1 mm in diameter produced by some Staphylococcus species. Circular shape of the colony, Trisobacterium indologenes. Filamentous shape of the colony, Bacillus mycoides with long irregularly placed or interwoven threads. Rhizoid with Flavobacterium columnare as an example that are irregular branch or root-like in character. For the next one, spindle-shaped colonies, and lastly, irregular shape of Mycobacterium tuberculosis.
Size describes how large or small bacteria colony is in millimeters. For the size of colonies, we have three. First, we have the pinpoint or small. This colony has less than 1 mm. Second, we have pinhead or the medium. This colony is about 1 mm. And lastly, we have matchhead or the large that has greater than 1 mm size. So for the last slide about size of colonies is for the easier understanding and visualization of the colony sizes. Here we can see the differences of the size. Next, we have margins of colonies, which is the edge or border of the bacteria or fungi colony. It may be important characteristics in identifying an organism. So we have 6 common margins of colonies. First, we have entire. It has smooth and does not have any notches. Second is undulate. It has wavy and shallow indentations. Third is serrate. It has irregularly notched. Fourth is filamentous. Long, irregularly placed or interwoven threads. Fifth is the curled, parallel change in a wavy strands. Last, we have lobate, with wavy and deeper indentions. So again, for the last slide of margin of colonies, it is for the easier understanding and visualization of margins, wherein you can compare them one by one through the image provided. For the elevation of bacterial colony, It gives information about how much the colony rises above the agar, and this describes the side view of colony. So we also have 6 common elevation of colonies. First, we have a flat, means level with no high hills or another raised parts. And raised, thick with a burp or terrace edges. Ambanate, raised but with a thicker center than margins, just like mountains. Convex, segment of a circle but flattened. Effuse, thin, veiny, unusual spreading. And lastly, we have subsurface. For the order of colonies, microorganisms can produce different types of volatile compounds that may give characteristic smells, pleasant smell or unpleasant scent. The production of these volatile chemicals depend on the particular organism's metabolic factor. Sometimes odor produced by this microorganism may give essential clues in the identification of microorganisms. For example, foul-smelling diarrhea and the absence of a blood or mucus in the stool distinguish gargiasis from bacterial or viral diarrhea. Microorganisms do not have odor on their own, but they may produce various metabolites that have distinguished smell of their own. Unpleasant smell. Anaerobes are particularly have unpleasant smell due to their reliance on sulfur hydryl compound to maintain redox balance. So here we have 5 examples of microbes and their smell. First, we have yeast-like smell. The colonies of Candida albicans have a distinctive yeast smell. Second, it has smells like soil or the soil-like smell. Certainly, cyanobacteria and actinomides synthesis geosmin, a volatile chemical reminiscent of potting soil. Rotten fish-like smell, we have Proteus mirabilis, produces a very distinct fishy odor. On Salmonella shigella agar, Proteus usually smell like rotten cooked fish. And bleach-like odor, we have the family of Carodens, a gram-negative rod responsible for wound infection. It gives bleach-like odor when grown in blood agar or chocolate agar. And lastly, the caramel odor. Streptococcus miliari produces diacetyl or the caramel odor. The detection of this caramel odor can be used in presumptive identification of Streptococcus miliari group. And for the impart pigment to surrounding medium, note color of surrounding medium. The main source of natural pigment are plants or microorganisms. The use of plant pigments have main drawbacks such as a non-availability throughout the year and pigment stability and solution. Large-scale plant use may lead to loss of valuable species. For this reason, the process may not consider viable microorganisms such as fungi and bacteria provide a readily available alternate source of natural derived pigment. Bacterial and fungal pigment have extensive application and have enormous advantage over plant pigments, including easy and rapid growth in low-cost medium, easy processing, and growth that is independent of weather condition. So we have examples here. Number 1, we have filamentous fungi are known to produce an extraordinary range of pigments such as carotenoid, melanin, flavins, penazines, quinones, monazines, violacins, and indigo. The use of monascus for angkak or the red mold rice production is the oldest record use of fungal pigment. So monascus produce yellow or the monazine and orange and purple pigment which are often encountered in oriental foods, especially in southern China, Japan, and Southeast Asia. Apart from plants, marine fungi also associate with the algae and coral. Reports suggest that marine endorphitic fungi produce pigment that help to mimic and often increase the beauty of associated life form. So fungal pigments are stable and various temperatures. So fungal and bacterial pigments studied or applied for commercial production. So here's the example of fungi, color, and pigment. So under fungi bacteria, we have monascus species for the color yellow and the pigment of ancapflavine. And number 2, we also have monascus species orange and rubropronactin and so on.
Now, for the second part, is the characterization of bacterial culture in agar's lands, and it will be presented by me, Elijah Nang. And so, examination of agar's lands culture must include the following. So the first one is the amount of growth. The amount of growth has four different results. So the first one is scanty or slight. The second one is moderate growth. The third one is abundant, which means a lot of amount of growth. And the fourth one is none or complete, which is completely no growth. So the second part is the color of colony. Now, a lot in agar-stands, there's a lot of colors. Usually it's yellow, pink, purple, or red, and this is caused by pigmentation. Many bacteria produce intracellular pigments, which cause their colonies to appear a distinct color. So a lot of different bacteria yield to different colors. Now, the second part, or the third part, is consistency. Now, there are four characteristics of consistency, and the first one is bacterius, which is butter-like. As we can see on the picture, it's very butter-like in color and in consistency. Second would be viscid. Its growth follows needles when withdrawn. The next one is membranous, which is thin, coherent, and it's like a membrane. The fourth one and the last one of consistency is brittle, which is dry and friable. The next characteristic would be optical characteristic. And so moving on to the first is opaque, which does not permit light to pass through. Translucent, some light passes through, but not enough to permit visibility of objects viewed through growth. Third one is opalescent. It's like an opal in color. And fourth one is iridescent, which exhibits changing rainbow colors in reflected light, which is rather cool. The fifth one is fluorescent, is having one color by reflected light and another by transmitted light. Second to the last would be dull, which means it's not glossy and it's quite boring. The last one is glistening, which is glossy. So another characteristic would be form. The first one is filiform. It's thread-like, and it separates or confluent colonies all throughout line of inoculation. Second is effuse or spreading. Its growth extends in form along the line of inoculation. C is accumulate or it's spiny. It's toothed or pointed in margins. Fourth is beaded, which is separate or confluent colonies at about the half end of inoculation. E, or second to the last, is rhizoid. It's irregular, branched, or root-like in appearance. And last is arborescent. It's branched and tree-like in growth, as you can see in the picture. Now, for the odor of agar plants, it usually resembles to a fruity odor. And so why does some bacterial culture have odor? So some microbes produce a characteristic smell, which sometimes help in identifying microbes. So the actinomides produce actually an earthy odor, which is often experienced after a first shower or rain. And many fungi produce a fruity smell, while E. coli produces a fecal odor. Now, medium and change in agar plants is usually from reddish orange to yellow. In triple irons, agar solutions, during preparations, tubes containing molten agar are angled, and so the slant of the medium is aerobic, while the deep or butt is anaerobic. When any of the carbohydrates are fermented, the drop in the pH level will cause the medium to change from reddish orange to yellow. From reddish orange is the original color to yellow. So that concludes the characteristics of agar slants.
Now, let me discuss the characterization of bacterial culture in nutrient broth. First is the surface growth. These growth patterns can be characteristic of an organism and may be used to aid in identifying an unknown organism. However, you must be careful when using growth patterns in broth to identify an organism because the type of broth and the temperature at which the organism is grown can alter the growth patterns. Also, if something happens to shake or otherwise disturb the tube, the growth pattern may be disrupted and subsequent observation will yield erroneous results. Under the surface growth, we have ring. It has a ring at upper margin adherent to glass. An example to this would be the Bacillus thuringiensis. Second is pellicle. It is growth in the form of a continuous or uninterrupted sheath over the surface. The example would be Mycobacterium smegmatis. Next is the flocculent. It is small masses of growth of various shapes floating in the culture fluid. The example to this is Bacillus subtilis. Next is the membranous. It is a thin coherent growth like a membrane. Second characteristics under the bacterial culture in nutrient broth is the subsurface growth, wherein different organisms exhibit varying growth characteristics. First is turbid. It is cloudy. The example to this is Staphylococcus epidermidis. Next is granular. It has small particles. Example to this are Streptococcus species. Next is flocculent. It has small masses of various shapes. Example to this is Saccharomyces cerevisiae. Also, there's a difference between the granular and flocculent. In flocculent, it promotes the clumping of small particulate matter, such as bacterial cell, while granular are that shows up as small spots. And the last characteristics under subsurface growth would be the flaky. It has large particles. Example to this would be the Mycobacterium fortuitum. The third characteristics of the bacterial culture in nutrient broth is the amount of growth. Here, we can see how much bacteria that were cultured. First is none or no growth. Second is scanty or small amount of growth. Third is moderate or average amount of growth. And lastly, abundant or huge amount of growth. The fourth characteristics is sediment. It is any particulate matter that can be transported and deposited as solid particles. Under the sediment, we have granular. It has small particles. Example to this would be Streptococcus species. Next is flocculent. It has small masses of growth of various shapes. Example to this is Saccharomyces cerevisiae. Next is flaky. It has large particles. Example to this is Mycobacterium fortuitum. Last, under the sediment, we have viscid. It is sediment rises as a coherent swirl. And last characteristics of bacterial culture in nutrient broth would be the pigmentation. It is a characteristic that is common to many species of bacteria. It also plays a role in adapting to different environmental conditions. Under the pigmentation, we have red. If the bacteria are grown at a temperature in which the enzymes responsible for synthesis of prodigiosin are produced, the pigment will be present and the bacteria will appear red in color. Example to this would be the Serratia marcescens.
Let's talk about the characterization of bacterial culture in Eger's lands. Under the characterization of bacterial culture in Eger's lands, we have three, and the first one is the amount of growth. Under the amount of growth, we have no growth. As you can see on the image, microbes didn't exhibit any kind of growth on the Eger's land. The second one is the scanty growth. In this amount of growth, microbes exhibited a growth on the Eger's land but only small amount. The third one is the moderate growth, and the last one is the abundant growth. As you can see on the image, microbes exhibited a very abundant growth on the Eger's land. And the second one is the form of growth. Under the form of growth, we have filiform. Filiform is a continuous thread-like growth with smooth edges. The next one is the beaded. Beaded is a non-confluent to semi-confluent colonies. The next one is the papillate. We also have villus. And the last one for form of growth is arborescent. As you can see on the image, arborescent is a tree-like growth. And the last one for the characterization of bacterial culture in Eger's land is liquefaction. Under the liquefaction, we have crateriform. Crateriform is a liquefied surface area is saucer-shaped. And the second one is the napiform. Napiform is a bulbous-shaped liquefaction at surface. The third one is the infundibiliform. Infundibiliform is characterized by its funnel shape. The next one is the saccate. It is elongated and tubular. And the last one for liquefaction is the stratiform. The stratiform is a complete liquefaction of the upper half of the medium. And the last part for this lab exercise are the study questions. And the first question is, why is it important to note all the cultural characteristics in the identification of bacteria? Our answer is, it is essential to note all the cultural characteristics of microorganisms because these characteristics serves as a basis for separating microorganisms into taxonomic groups. The second question is, are cultural characteristics enough to identify a certain genus or species of bacteria? And our answer is, cultural characteristics only provide additional information when it comes in identifying a bacterium. That means, cultural characteristics are not enough because in identifying a bacteria, biochemical and motility tests should be also considered. And the last question is, which of the cultural characteristics are subject to mutation? Our answer is surface properties and margin.